Hi, my name is Björn Högberg, and I will share some quick thoughts on viruses and how I believe we should start fighting virus pandemics in a new way before even we have vaccines or to complement vaccines. So, what are viruses exactly? Viruses are, in principle, just nanoparticles that carry some genetic information. And this genetic information is either RNA or DNA, which are two forms of nucleic acids. In the case of the SARS-CoV-2 virus or the Covid19 virus, this is RNA. But in the case of, for example, herpes viruses, these carry DNA inside. And to make it simple, I will just call these molecules the virus genome. And that could be either RNA or DNA. It doesn't really matter for the rest of this discussion. So a virus enters the body, and then they attach to cells by binding to molecules on the cell surface. And then they enter the cell, and there, inside the cell, they dump their genome into the cell, and the cell, not knowing better, starts to use the genetic information from the virus as if it was its own. So this malicious code from the virus hijacks the machinery of our cells and use it to make new viruses. So proteins in a cell are basically the machines of the cells. And these proteins are fabricated from component molecules inside the cell, and they are assembled in a specific order according to a gene. And in the case of the virus genome contains information for the cell how to create new virus proteins. And when these virus proteins get started, these new virus proteins in turn start to copy the virus genome. And they also start to making new component proteins for the virus particle or the shell. So while the shell is going about its normal business of making proteins for itself, it is now also making proteins for the virus. And this is essentially all a virus is doing. It enters cells and dump their DNA or RNA genome, and then let that sit and make copies of itself and all the components for the shell. And when everything is assembled together, they form new virus particles and have the genome inside, and they leave the cell to infect another cell and replicate again. And this is all a virus does. It makes copies of itself using hijacked cells. And all a virus is a shell with some DNA or RNA genes packed inside. So for all intents and purposes, the virus genome, its code, is the virus. And this code is just making copies of itself. And I think it's important to remember that at its core, all viruses are just this simple algorithm, self-copy. Of course, the implications for the host, the disease that they can cause, can be really complex. But the basic premise that all the viruses share is this simple algorithm. So how do we stop a virus from spreading? The immune system does this in two main ways. One way is to develop binder molecules or antibodies that bind to the virus. And because of these bound antibodies, it makes it impossible for the virus to bind to cells and thereby stopping the replication. Another way, this doesn't take care of the virus factories lying around. And the virus factories, in this case, are of course the cells that are already infected. If the immune system manages to do its job well, these infected cells will eventually be detected by killer T cells, and these killer T cells will induce apoptosis in the infective cells, basically telling them to shut down and die, which they will do. You'll notice here that these methods that the immune system is using are not really destroying the viruses themselves. They're only stopping them from reproducing. As a side note here, actually the virus particles themselves will eventually be destroyed in the body, but that is more of a general cleanup mechanism. The main way of stopping the virus infection is to immediately kill off its ways of reproducing. So what I think, the immune system stops viruses by blocking their replication. And I think we as a society can do the same thing. We can stop any virus by blocking their path to reproduction. And the analogy with T cells is, of course, that if we are able to detect an infected individual, we could tell that person to shut down. I mean, we wouldn't, of course, tell the person to shut down completely, but just for a week or so, we could basically tell them to isolate and quarantine. And this is basically an immune system for society. All we need to do is to detect these people. And I think it's important here to remember that viruses are basically a very simple self-copy algorithm. In my opinion, we, as a knowledge-based society, should be able to come up with a really basic, simple strategy to counter them. And the strategy, of course, is to test everyone continuously and then ask the people who test positive to stay at home. And please note that I'm not talking about mass laboratory testing schemes by healthcare professionals, contact tracing or surveillance. This strategy that has been proposed by several of my colleagues, please see the links and references in the description. This strategy is really simple. Let everyone test themselves in their homes a lot and then stay home if they get a positive test. Wait a minute, you say. We can test ourselves in our homes for COVID-19? Yes, it works right now. The tests are just worse at detecting really small amounts of viruses. So for some reason, a lot of people ignore them. I will talk more about that later and why this is not a problem for this strategy. But for now, let me tell you how these types of tests work. The tests that I'm talking about here are mainly the ones called rapid antigen tests. There are many other really promising rapid tests being developed, but here I focus on the rapid antigen tests, as these tests are the ones that are most widely available right now. Why are they called antigen tests? Because they detect virus proteins. If the virus proteins were in our bodies, we would slowly start to develop antibodies for them. And stuff that antibodies bind to are called antigens. It is a bit confusing, but that is how it is. To take one of these tests, you typically take a swab sample from your throat or nose, and then you dip the swab into a solution that collects the sample in liquid form and typically breaks up the viruses. Then you pour some drops of these onto a lateral flow device, or LFD, sometimes also called lateral flow assay or test. And this is basically a paper strip test similar to a pregnancy test. The drop of solution breaks up the virus particles, and then the solution, virus proteins, and lots of other stuff from the sample will start to migrate through the paper due to capillary forces. The first stop of the liquid is the so-called conjugate pad, and this first stop is a depot of gold nanoparticles that are decorated with antibodies that bind to virus proteins, that is, antigens. So if there are virus proteins in the solutions, these will bind to the gold particles via the antibodies, and these are loose, so they will continue to move. The gold particles will continue to move through the test strip. The second line here is the test line, and these are antibodies that are bound to the paper. And the third stop is the control line. Here are antibodies that attach to the antibodies on the gold particles themselves. And let's have a closer look and see what happens if there are virus proteins or not. If there are virus proteins in the sample, the gold nanoparticles in the conjugate pad would start binding these and get dragged on through the paper strip. And when these gold particles enter the test line, some of them will get stuck because the antibodies on the test line will bind to the virus proteins as well. So some of the gold particles will now be stuck here. And the control line will catch gold particles no matter whether they bind virus proteins or not, because the antibodies there in the control line will bind to the antibodies on the gold particles themselves. And the nice thing about gold nanoparticles is that they catch and reflect a lot of light. So if your sample contains virus proteins, you will see two purple lines, one at the test line and one at the control line. If your sample was empty of virus proteins, that is antigens, most of the gold particles will bind to the control line only. So you'll see only one purple line. If this line doesn't show up, then something was wrong with the test and you need to redo it. And this is how rapid antigen tests work. The whole process takes about 10-15 minutes. And the nice thing about these tests is that they are easy to develop for a new disease, making the antibodies that are required for these tests can be done very quickly compared to the time it takes to develop vaccines. What would be really great though would be a test like this that detects the virus code instead of the proteins, so the genome directly. There are some of these under development, so maybe we'll have them soon. But that would actually be amazing because we typically know the virus genome like a week after a new virus has been discovered. And if the test is a test that can detect RNA or DNA, which we know are in all viruses, adapting them for a new code of RNA or DNA would be a piece of cake, like really simple and really fast. But for now though, we have really good rapid antigen tests and they work for COVID-19. So the simple algorithm is this. Let everyone test themselves at home repeatedly and ask people to stay at home for at least a week after their last positive test. And the testing interval should be small enough to catch the most infectious stage. For COVID-19 testing, every fourth day would most likely be enough. So testing everyone every fourth day and ask the ones that get a positive result to stay at home for 1 to 2 weeks. Is that feasible? Well, I think there are several things that are needed to make this work. First of all, the tests absolutely need to be at-home tests without need for medical personnel. The rapid antigen tests that are out there today can be taken as a self-test. They might not be approved for it everywhere yet, but believe me, anyone who can follow simple instructions and who actually cares about testing properly will do these tests almost as good as a pro. Now, there are some indications from a study in the UK that healthcare professionals are slightly better at making the tests work. But in my opinion, and several researchers agree with this, the benefits of at-home testing far outweighs the small drop in efficacy self-testing gives. And more on this a bit later. So what about test quality? These tests are not very good, right? Well, it's actually not true. The tests are good enough, really good for what we need here actually, which is detecting infectious individuals. So note that the purpose of this simple algorithm is not to give everyone a 100% answer on whether or not they might have COVID or something else, but to limit the spread. These tests will not be the deciding factor for clinical decisions about your healthcare. For that, you will take a lab test if needed. The only decision we need to make from these tests is whether or not you should stay at home. So as you will see in the following slides, the test needs to be good enough, but it doesn't need to be as good as a clinical test. So what if people don't comply with this? You probably know some people who don't think COVID is anything we should care about, right? But for this to work, people need to follow the instructions, right? And as I will show you, we can get by with far less than 100% compliance for this strategy. So let's look at this a little bit more carefully. You've probably heard about the R value. This is the number of people that one infectious individual on average will infect. So if you have 3 infectious individuals and an R0 value of say 2.4, these 3 people will on average infect 7 new people. And if people go on living like usual, so the R value doesn't change, it will still be 2, 4, and 4, these 7 newly infected will go on and infect on average 16 new individuals. Okay, so now let's look at a situation if all people that are infected quarantine themselves. If people self-isolate, they will infect much fewer people on average. They will still probably infect more than 0 individuals, at least on average. Some may infect people at home, for example. But let's say that each quarantined individual, on average, infect about 0.3 persons. So let's call this new R value RQ for quarantined, as opposed to R0, which would be for people going about as normal. And if we have 9 infected individuals that are self-isolating, and the R value would be RQ equals 0.3, then these 9 would infect about 3 people. And then these 3 would infect 1 on average, and if it continues, there will soon be 0 infected people. As I said earlier, we cannot count on everyone that is infectious to self-isolate. There will be people who don't care, and there will be people who get the wrong test result. So what we have to do is to look at a mixture of free-moving and quarantined individuals. Let's assume that 2/3 of the people that are infected will self-isolate. So if we had 9 infected individuals in total. 6 of these would self-isolate, 2/3, and 3 would continue as normal. These 3 people that don't self-isolate will continue to spread the disease with an R value of 2.4 as before, leading to 7 newly infected individuals. But the 6 people who self-isolated, they will only spread the disease to 0.3 times 6, that is 2 individuals. So in that case, we will have 7 infected, 3 from the high spreaders and 2 from the 6 low spreaders, giving us 9 in total. So when we go on to the next time point in this series, there will still be only 9 infected individuals. Again, let's assume that 2/3 of these 9 individuals go on to self-quarantine. That will be 6 self-isolated and 3 high spreaders left. Again, the 3 high spreaders will spread to 7 individuals and the 6 self-isolated will spread to 2. So in total, there will still be on average 9 infected individuals. So you can see that if we manage to put enough of the newly infected in isolation, we can control the spread. Imagine if we, instead of 2/3, put slightly more than that in isolation in each step. Then the disease would die out. So how large a proportion do we need to accurately detect and persuade to self-isolate to kill off the viral disease? Let's do the math. So we call R0 the natural infectivity rate and RQ the infectivity rate of the isolated individuals. And let's say that X is the percentage or fraction of infected people that both accurately test and self-isolate. Then the overall apparent R value, or R total, would be X times RQ plus whatever is left of the other non-isolated individuals, which would be -1 minus X times R0. To actually suppress the spread, this R total needs to be lower than 1. So if we know the overall R value, if we know that that value needs to be lower than 1, we can take this expression for R total and solve for X. And then we see that the proportion of detected and isolated cases X should be higher than R0 minus 1 divided by R0 minus RQ. And this simple model was proposed quite early in the pandemic by some of my colleagues, and you can read their paper online. Links are in the description. Okay, now we come to the part where we look at what test quality we need and what compliance we need. This fraction X of isolated cases is actually just the product of the percentage of compliant individuals and the percentage of the time the test gives us true positive results. So let's look at this. Let's say we have 13 infected individuals. Out of these 13, only 75% are willing to take the test and self-isolate if they are positive. So out of these 13 individuals, 10 will comply and 3 will invent some bad excuse for why they shouldn't have to. And in this case, the compliance, or C, would be 0.75. Okay, let's assume that the test is imperfect and misses some of the actually infected individuals. Let's say that out of the 10 people that complied and tested, only 8 of them will get a positive test result. So 2 of these guys will get false negative results because of bad test or malhandling of the test somehow. And here, P would be the true positive rate, and in this example it would be 80% or 0.8. So in total, if you look at both compliance, C, and true positive rate, P, only a fraction, P times C, will actually go on to self-isolate. And the remaining fraction, 1 minus PC, or P times C, will go on and continue as normal. So you can see here that the fraction X that we got earlier is just the product, P times C, the efficacy of the test and the compliance taken together. So for this strategy to work, the combination of the efficiency of the test and the compliance of people actually to go through with it needs to be high enough. And according to this simple model, P times C needs to be higher than R naught minus 1 divided by R naught minus RQ. So let's use some commonly quoted figures for COVID-19 as an example. Reasonable estimates for COVID-19 should be that R naught is around 2.4 and that RQ could be around 0.3. Using these numbers, we get that the product of compliance and test efficacy should be higher than around 2/3 or 66% for this strategy to work. Of course, note that this is a simple model and reality is of course much more complex, but I think it's a good understandable explanation of why the foundations of this strategy work and how it actually could work altogether. I think it shows that for the test everyone strategy to work, compliance and efficacy most likely do not need to be 100%. They just need to reach a certain threshold. And the threshold is related to the natural infectivity rate and the quarantined infectivity rate. And of course, the natural infectivity rate here can of course be pushed down using other measures if necessary, such as banning large parties and mass gatherings and things like that. So even if you think these numbers for infectivity rate, if you think that they are too low, we could still use this strategy in combination with other measures if needed. But let's assume for now that these values are approximately correct for COVID-19. If that would be the case, are the tests good enough and what level of compliance do we actually need? These rapid antigen tests have now been used in quite large studies, so we know a great deal about them already. And here are some data from a recent study in the UK, where they used rapid tests on quite a large number of individuals and then compared the results to laboratory PCR tests. So the bars show in gray here how many individuals tested positive in a laboratory PCR setting. And this type of testing gives quantitative estimates of the exact number of copies of the viral genome found in the patient samples. And this is what is shown on the x-axis here, the bottom axis. So the RNA copies per milliliter can be written out directly, or it can be written as a so-called CT value from the PCR test, or written as the 10th logarithm. And these three ways of writing the numbers of genomes are equivalent. So for each group of people, grouped here on how many viral genomes that you find in the samples, the red bars show how many of these also tested positive using rapid antigen tests. And the proportion of red versus gray is this line here, plotted on the right axis here. And you see that for individuals with low numbers of detectable RNA copies, the rapid antigen test gives positive results only in a small fraction of all PCR tested individuals. For example, if you look at this bar here, only about 10% of this group gives a positive result on the antigen tests compared to the PCR test. So this doesn't look very good, does it? Well, remember, to spread the disease, an individual needs to be an effective virus factory. So we're really only interested in catching the individuals that will actually spread the disease. So more and more research is now coming out that indicate that these groups are very likely not very contagious, if at all. So if we were able to isolate replicating virus from these groups, but not from these groups, then that would corroborate that we can safely ignore these groups for our test all strategy. Luckily, there are such experiments published already. In this recent study from the Netherlands, researchers took patient samples and then measured the amount of detectable virus genomes and tried to culture viruses from the same samples. The black squares indicate where they were able to culture virus and see active replication. all the other circles here are samples where they could detect pieces of the viral genome using PCR, but they couldn't see viral replication. I say pieces here because the PCR tests we use for COVID-19, it only detects a small region of the virus genome. So each hit in a PCR reaction could be an entire genome, but it could also just be small pieces of it that are, for example, leftovers that aren't yet cleared by the body after the infectious stage. So if you look at the x-axis here, these are the same type of values we had in the previous graph. So if you compare these figures, you get, in my opinion, a good indication that these groups are the ones that we have to worry about when it comes to infectiousness, and not these groups. Here are more indications of the same thing from another recently published study. And this is a cohort study where they have followed transmission chains of individuals during the initial wave in Spain in the spring of 2020. And these cases had contact tracing data, so the authors were able to identify how many of the contacts eventually ended up getting COVID-19. And what they found is this, that the people with a high number of RNA copies detected by PCR had a higher probability of transmitting the disease. And in this Spanish study, they found that 90% of all transmissions were from individuals with an RNA load higher than log 5 copies. So that's in this group here and above. So if you take all of this data together, I think it makes a compelling case that rapid antigen tests probably catches a lot of the actually infectious individuals. Probably it can catch around 94% of actually infectious individuals if you look at these data. It's only if you also count these groups that the rapid antigen tests appear unreliable, but for assessing the probability of infectiousness, these tests are actually really good. So in our equations from earlier, we could probably put p equals 0.94 here. With this, our compliance C could be as low as 75%, for example, and our C times P would be well above 0.66. If these data are wrong, and these tests turn out to be a bit worse in practical use, we could most likely still make it work by pushing up compliance and other measures. So the take-home message after all of this is that the tests we have right now are perfectly good for the purpose of assessing infectiousness. And that is essentially all that these tests need to do. We already have perfectly good tests for clinical decisions. And also, the argument that unless everyone follows the testing regime, the strategy will fail is false. We just need enough people to follow, not everyone. And in total, I think this can be an extremely powerful method of suppressing the spread of any infectious disease. So what would it cost though? Well, these tests can be bought right now for about €5 a piece. I just saw that Austria actually bought a ton of these for almost half that price. But okay, let's assume that they are still around €5 a piece. And let's assume that we set ourselves for 6 months in total every 4th day. That's around 46 tests per person. So if we have 10 million people times 46 times 5, that was around €2 billion. Okay, not super cheap, but way cheaper than what COVID-19 is costing right now. And to put that in perspective, for example, in Sweden, a country of 10 million people, we've already spent more than 10 times that amount on economic relief packages, something that would barely be needed if we would have used this strategy from the beginning. There are, of course, challenges with this approach. As with any new population scale technology, it will take time to perfect. But I really think we have to try this now. If you leave a virus long enough time to replicate, it will perfect itself by mutation, which is exactly what we're seeing right now. A rapid testing strategy could end this now, before the vaccines do, and could have ended it earlier if we would have had the knowledge already in the spring. And even if you do this half-heartedly, it will still lower the R value without imposing further restrictions of freedom of meeting and traveling. And I think in the end, one really important overlooked point is that if this works, it will give us a really important knowledge for the next pandemic, giving us the tools to stop it even before it becomes a pandemic. So we have the ability to make vaccines, of course, but until we learn how to make new vaccines within weeks, we need new ways to counter viral diseases. So I hope you've learned something, and if you have questions or comments, please contact me via Twitter. In the video description, I've left some links and references for most of what I've talked about here. If you're interested about this stuff, you should also look up Michael Minna from Harvard, who has worked a lot on these issues and greatly inspired this talk. Have a good day and happy rapid testing.